All right, well, welcome everyone to this presentation. My name's Darren Morton. I'm the Director of the Lifestyle Medicine, Health and Wellbeing Research Centre here at Avondale University College. And I work very closely with Mel Renfrew, who's the course coordinator for postgraduate studies in lifestyle medicine. And the unit one of the units that's being offered this semester is called Physiological Underpinnings of Chronic Disease. And we have a really stellar lineup of our guest lecturers. And you're going to hear from the first of them tonight, Dr Joanna McMillan. In about a month's time, we have Professor Gary Egger, who will be presenting a module on metaflammation. If you've never heard of that, well, then you might like to join us as well. And then later on in the semester, in a couple of months' time, we have Dr Cameron McDonald, who's going to be presenting on personalised medicine. And you'll really like him. I don't know if this made him feel bad about himself, but one day we were at a conference and I said, Cam, you remind me of a younger, better looking, more intelligent version of myself. And so I don't know if that offended him or not, but anyway, he's a great guy. So you really enjoy hearing from Cam. But tonight we are very, very privileged. We've got Dr. Joanna McMillan, as you can see there on her first slide. You know, I actually, when we had this module, in the course, the module we were looking at was the gut microbiome. And I was talking with Mal, the course coordinator, and we said, you know what, let's just put it out there. Let's just go for gold and get try for the very best person we could imagine to teach a unit like this. And so I emailed Jo, and then she came back and said, yeah, I'd love to be involved. And I'm like, so I got on the phone. I haven't told you this, Jo. I got on to Mal and said, guess what? And like, we all had a little sort of happy dance. So we're very, very excited that you're part of it. And I'll tell you why. Jo comes from Scotland. You're going to pick up that probably subtly, at least, in just a moment when she starts presenting. That was where she did her undergraduate degree, but then did her PhD at Sydney University. And since that time has really, look, I think it would be fair to say, become perhaps the most trusted dietitian, food expert in the popular media circles in Australia. She's done a lot of media work. You'll see. I'm sure you've seen her before. For those that are in this class, one of your tasks that we wanted you to engage with was the Catalyst series that Jo featured on, which was fantastic. And look, I've seen Jo present many, many times. Every time I learn something new, which I really appreciate. But I think one thing that probably I most appreciate, Jo, about what you do is you have a very, very evidence-based but balanced approach to nutrition. And I think, for some, like I come from more of an exercise background, and no one gets that animated about what type of exercise we should definitely be doing and you shouldn't be doing that one. But there's something about food that just polarizes people. And I really like Dr. Joanna's just approach to it. I think it's, I just really appreciate that about what you do. So anyway, I'm going to hand it over to you. Thanks so much for being with us. And yeah, we look forward to hearing what you have to share. If you have a question, just type it in to me and then I'll sort of coordinate that at the end. And in a couple of weeks' time, we'll be meeting with Jo again, where it will be more of a Q&A format. So tonight she'll actually be presenting a formal presentation. So, Dr. Joanna, thank you for being with us. Take it away.
Cool. Thank you, Darren, and thank you all for joining me at 7 o'clock on a Monday night. I hope your brains are still awake. Look, this is a really fascinating subject. It's a subject I've really immersed myself in over the last couple of years, starting with the Catalyst Gut Revolution show. And then sort of that sparked my interest in so many areas that I felt I wanted to follow up more on. And what was really interesting for me was the fact that diet and lifestyle, which Darren, myself, Gary Egger, Cam McDonald, the other speakers you're going to hear from over the coming weeks, all of us are immersed in this area of lifestyle medicine, all we're coming from slightly different angles. But what is fascinating about understanding the microbiome is that not only is it intimately involved with overall gut health, but it really is central to so many aspects of our physical and mental health. And it is so influenced, I would say the major influencer is your diet and your lifestyle. So this is an area that we have to know about. Anyone involved in health, anyone involved in physical or mental health has to try to have some level of understanding of this area. fast moving area. I think when you start to read more about this, nobody can be on top of all of the studies because there are literally so many coming out every week, that it's a good thing, I think, to just sort of find yourself looking at the broad area. Make sure you read widely, because if you only read sort of specific little one studies, then you might get sort of drawn down into the rabbit warren and not understand what the big picture is. So what my intention is tonight is to really just give you that big overview to understand what is it that you need to know, how does the microbiome develop, and what are the major influences. So obviously my key area is nutrition, but I've also worked in exercise. That's kind of where I started my career. And I've got a huge interest in the whole of lifestyle medicine. So I may just sort of touch in and throw in a few of those areas, but we are going to focus mostly on the microbiome itself, what you can measure, and how food and your lifestyle influences that microbiome. So let's have a think first of all about why is everyone so excited about the microbiome? What exactly is it that it does? Well, it does a whole number of different things, and the immune function links are what are so interesting right now. Of course, one of the things that is dominating the news is the coronavirus, and of course I'm being asked, is there anything we can do from a nutrition perspective and from a lifestyle medicine perspective to make ourselves stronger, so that if the predictions are right and that the vast majority of us are probably going to come across this virus at some point, how are we best able to fight it? And that's what's really worthy of remembering. I think this is something people forget about. We see kind of blasé articles online that talk about, oh, the foods that are best to boost immunity. But the truth is that it's broader than any individual food. Yes, we need certain nutrients. I've got an article up on my website at the moment that is about the nutrients you need for a good, strong immune system. But the truth of it is that the bulk of our immune system, because the gut is really, the gut and the skin are our sort of outside barriers. They're the first line defense against any invading viruses, bacteria, other pathogens. And so it should not be surprising that about 70% of our immune system is actually centered in the gut. So the gut is a really important part of that. And the microbiome actually trains the immune system. So it does that very early in life. So that's one of the reasons we know it's very, very important in those first couple of years of life that infants develop a strong microbiome, because that microbiome is training the immune system to understand what are potential foreign objects that we need to mount a defense against, and what are normal proteins that are part of food, for example. So we're starting to see evidence that is linking the microbiome and the development with allergies in food and drinks, for example, with autoimmune diseases, and with immune function throughout life. So we're going to talk a little bit about how the microbiome develops. But then what's also fascinating is that it continues to be important. So your microbiome continues to be important throughout life because it is interacting with immune cells and with immune patches, pairs, patches throughout the gut all of the time. So rather than, I think one of the problems that we have is that we've kind of come through this phase since people like Louis Pasteur understood about how bacteria, viruses, other microorganisms can potentially be harmful. We've become really sort of germophobic. So we kind of had this idea of everything has to be antibacterial, and we're seeing it right now with coronavirus, everyone bleaching their houses and rubbing antibacterial gel all over their hands without really understanding exactly how microbes work. So instead of understanding that the vast, I put a post up lately, the stat off the top of my head, it was something like 95% of microbes are actually either neutral towards us or they're actually beneficial compared with the ones that are pathogens. And that's what's worth remembering when it comes down to our gut microbiome. And I might just explain the difference in those words. Microbiota are the actual bugs. The microbiome is the genetic material of all of those bugs. So it's the bugs and their genetic material. So I'll try to use the word microbiome as much as possible because that is the functioning of the microbiome overall that's ultimately important. And I'll come back to that. So the bugs that are in your system actually are there largely helping us. Every now and again, we come across a pathogen. Probably everybody listening has had a gastro bug at some point. We know how unpleasant that can be. But the vast majority of those hundreds of species of bugs that are living in there are either just there, not harming us, just living in synergy with us, while as the vast majority of them are actually helping us. So they're interacting with our immune system and they're very important there, but they also help us with digestion. So we are built, one of the constant debates I'm asked to talk on is, should we be paleo or should we be vegan? Are we omnivores, or, you know, are we carnivores? We've even got people who refuse to eat plants these days. We've got the omnivores, we've got the vegetarians, and we've got the vegans. And all along that spectrum, the bottom line is, if we just look at our physiology, the way our gut is built and the microbiome in there is built to digest both animal foods and plants. Now, that doesn't mean you have to have both. I would argue that you have to have plants. I'm certainly not going to adhere to the carnivores, but if you want to be vegan, of course you can survive that way. But our gut physiology and the microbiome tells us that, in fact, we're designed to be able to break down both. And the microbiome is particularly important for breaking down those tough plant foods. And we'll come back and talk more and more on those plant foods. That process is important, not just because it helps us to digest the plant foods, breaking down the stuff that our enzymes can't. It does produce some nutrients, so it produces things like folate. It produces vitamin K. In fact, most of our vitamin K comes from bacterial production, but it also produces a whole load of the byproducts of fermentation that are important. So one that we'll come back to is butyrate, and that is the major fuel for keeping the gut lining healthy. So you'll have heard in the past lots of stuff about leaky gut syndrome. It's not a term I like, but certainly now we know. It used to just be thought of as, that's a load of nonsense, it doesn't happen. Now we've got really strong evidence. We can actually view it. I've seen it with a gastroenterologist I worked with on the Catalyst show, where we can actually see that gut permeability is affected when the gut lining is affected. So depending on how healthy your microbiome is and the products that are being produced within the gut actually influences how much mucus is produced. It influences the turnover of cells. So it impacts on that, the ability of the gut to be permeable and to be a strong gut lining, letting in what it should let in and stopping what it shouldn't be letting in. Now, that doesn't mean that you get stuff floating up into your bloodstream, but what it does mean is that you can get bacterial translocation into some of those layers of the gut, and that of inflammation and other byproducts that then can reach up into the bloodstream. So I think it's really important to understand exactly what the gut barrier function is doing and what gut permeability really means, rather than getting too caught up in this term of leaky gut syndrome. So we know that metabolites produced from the bugs also travel to the heart, to the lungs, to the liver, the kidneys, and the brain. So you've probably heard, no doubt, about the gut-brain axis, but it's worth remembering that there is now talk of this gut-heart axis, the gut-liver axis, the gut-kidney axis. So we're starting to understand more and more and more about exactly how it is that what's happening in your gut is actually influencing all sorts of aspects of physical health. It's even influencing your blood sugar control, your insulin sensitivity. And then probably most out there is really trying to understand how it impacts on mental health. And we'll talk a little bit more about that. First of all, just how the microbiome develops, and because this sort of also gives us some key indicators for those of you who are involved in pediatric medicine, for example. This is something that is going to become more and more part of our future, understanding where we need to have an interaction and where we could potentially prove some benefit for someone's lifelong health by intervening a little bit sooner and helping them to develop a strong microbiome. Now, in the old days, we used to think that life inside the womb was completely sterile. And in fact, in recent years, that's shown to be not quite correct. So even while the young fetus or the infant is still in utero, there is actually already some microbes being in contact with them, and the microbiome is already starting to be impacted. But the mode of delivery, whether it's a vaginal delivery or whether it's a cesarean delivery, has a major impact. And that's not hard to understand. If you just think about a vaginal delivery, what sort of microbes is an infant first going to be exposed to? And of course, it's going to be exposed to microbes that are in the vaginal tract itself, but also in the gastrointestinal tract, before it then comes into contact with the skin. If you think about a baby that's delivered by cesarean, then they are not exposed to any of those microbes. They are coming out, and the first microbes they are exposed to is the skin of either the nurse or the doctor who delivers them, or often these days the baby is put onto the bare chest of either dad or mum. So that's a very different sort of, those are aerobic microbes for a start, that are living on the skin compared to the anaerobic organisms that are living deep inside of us. So you can see immediately there's a very different exposure. So we already see, and this is happening in a crude way in some delivery suites, where doctors are trying to somehow, you know, get exposure, and babies that have to be delivered by cesarean, or they're trying to expose them to some of the bugs that they would have done if they'd been delivered in a vaginal way. So, but I think that will become, it's fairly crude at the moment, I think that will become a little bit more, how should we say it, a little bit more organized in the way that it's delivered. And then once the baby's delivered, and this is an important part, you know, I think in the West, we do have an issue where lots of babies are delivered by cesarean when they need not be. I think women have to be given the choice but they need all of the information before they make the choice as to how their babies are delivered. Nevertheless, we're always going to have some babies delivered by cesarean because that saves lives of babies and mothers. So if you were delivered by cesarean, or your kids were delivered by cesarean, don't panic. It doesn't mean that you can't do anything about it, because actually that is just one little factor. What is probably even more important then is what happens from that point on. And even as an adult, you can make changes to your microbiome. That's what's pretty extraordinary about the microbiome. It reacts very quickly to changes in its environment. But let's continue with our infant. So we're moving into the infant year, whether the baby is breastfed or bottle fed. And of course, formulas have cottoned onto this. So we're seeing lots of infant formulas now with trying to replicate some of the oligosaccharides. These are the little short chain carbohydrates that are in breast milk that we know that our human enzymes cannot break down. So if that's not an immediate sign that the microbiome is important, then what is? We have these particular fuels that are there, these particular carbohydrates present in breast milk that are there to specifically feed these kinds of beneficial microbes. That's pretty extraordinary. So we now have formulas that are trying to replicate some of that. Of course, we can never truly replicate, because breast milk is also ever changing. It's very specific from the mother to the baby. But formulas have certainly got a whole lot better in terms of their delivery. We even have some formulas now with some probiotics in there, or with these specific prebiotics. So prebiotics fuel the good bugs, probiotics are giving the good bugs themselves. And then what happens, we've got what happens with their feeding, when are they weaned, what are they weaned with? It bothers me enormously that a large majority of babies in Australia are put onto a highly refined, ultra-processed rice milk with things like iron and other nutrients added to it. That's not a whole food. It's not what we need to be feeding babies. We need babies to get onto solid foods and whole foods and an array of different plant fibers. They can get their iron from meat. You know, we don't need to be fortifying. We could be giving them whole foods, as has been done until more recent years. But aside from that, we have the issue of antibiotics, we have the issue of the genetics, and we have the issue of environment. So there is a lot of work going on around whether babies are allowed to crawl around on the floor. I'm going to show you some data and a few slides of kids who have grown up in Africa compared to an urban developed country, and you see very different microbes there. So there is certainly an issue with, you know, some have sort of classified it as, are we too clean? So we have bleached floors and babies are antibacterial wiped, and all of this kind of hygiene is great for preventing pathogens, but it's unwittingly throwing the proverbial baby out with the bathwater. So I think we're going to understand a lot more about how can we ensure that kids have the best environment to build a strong microbiome. Antibiotics are of course life-saving. Doctors have become an awful lot better about knowing when antibiotics and when they need to be given, but sometimes they need to. You know, one of my sons had to have antibiotics at 3 months old for a pretty severe respiratory infection. So antibiotics do have to be given, but if we can understand more about how to help kids to recover from having antibiotics and how to support the microbiome as best we can, then that's the way for us to move forward. And then moving on, by the time the child is about 3 years of age, they have a more adult, or should have a more adult-like diet, and they have an adult-like microbiota by that stage. So those early years, sometimes it's called that, you know, the first 1,000 days or the golden 1,000 days, because those sort of conception to year 2 is exceptionally important. And I would extend that for the microbiome up till about age 3 is really, really important for the development of the microbiome. As I said, though, after that, we still have some major influences. Now, one of the things that has really stepped forward this research in huge strides over the last few years is the ability to measure it. So I'm going to say right up front, be careful with the tests that you're using. I'm going to talk about the test and the company that I am most familiar with and understand and know and have worked with. We use this company. They're researchers from the University of Queensland who have set up a commercial testing company. But they're not the only ones. So forgive me for seeming to be a little bit biased towards this company. I'm not in their payroll. It's just the company I'm familiar with. But what I do ask you to be hesitant of is looking out for the charlatans. You cannot measure the microbiome from a traditional stool culture test. That's very, very important. Stool culture tests still have a role. If you get a patient in who has a tummy bug coming back from holiday, it can still pick up pathogens, but it cannot measure the microbiome. So remember what I said about most of those bugs are anaerobic. They will not grow in a petri dish in the lab. So stool culture is not what does it. So these kind of modern tests are actually doing what's called metagenomic measuring, and what that is essentially measuring the genes of the microbes. So it is done from a tiny swab from used toilet paper, and that's what you send off, and you get this report that is what I like about Microba is the name of the company I use. What I like about what they're doing is that they then talk about this as being an insight report. So anyone who asks me about having their microbiome tested, I always say, just be careful. This is not yet diagnostic. It's not going to give you the answers to your irritable bowel syndrome or the gut problems that you've had for years. It is going to give you an insight, and it might help to direct you towards a healthier diet or including particular foods that might help you to improve your microbiome. But while we have a huge amount of knowledge about the microbiome, we're still learning. So it's important that some of the things that we see, we don't yet know whether they're simply associations, whether the bugs are just adjusting to the diet that's coming in, or whether they're actually causative of some particular factors. So I like the use of this word insights. It's giving insight, and it's helping build the research to understand more. So what you're seeing on the screen there is actually my report. This is actually from a couple of years ago. It's worth doing it fairly regularly, and you can compare your reports to see if your microbiome's changing over time. But this is mine from a couple of years ago. And you can see there that I'm bang in the middle of what is termed as the healthy group. So you can compare yourself to your age group, your gender, and so on, and look at exactly where you're lying. Now, if you're outside of that blue group, that you have an unhealthy microbiome. It just means that you're different the comparison group. So that's important to understand too. So actually below that, the little scale that's showing the diversity level is arguably the most important thing about your microbiome. So what we know, and when I was working on Catalyst, I interviewed experts from all. What's nice about this area, together people involved in nutrition, biology, microbiology, epidemiology, all sorts of different and diverse areas of science. And the one thing that they all agreed on was the most important thing about your microbiome is the diversity. So diversity is king. You're going to hear me talk about that in relation to diet, as well as in relation to your microbiome. So diversity means that you've got a really broad number of different species. And it's kind of a little bit like, I don't know, thinking of humans around the globe and thinking we do best when everyone's even. So there's this thing called evenness with your microbiome. If you have an evenness, that means nobody's dominating. So we don't have one culture dominating and another culture being suppressed. You have this really nice evenness. You've got lots of different groups, all doing different things and all different functions. That is the healthiest microbiome. And that's what we know to be the strongest predictor. You'll see then that you also then get these insights. I didn't have any major standout insights, but then these, whereas I've read hundreds of these different reports, a lot of people have specific insights that you can look at and give you some indications. So I'll tell you a little bit about some of the things that these insights are looking at. I hope I'm getting a little bit of a flash up saying my internet is sometimes unstable. So I promise I've kicked the teenagers off their devices. So hopefully you're hearing me okay without too much interruption. So let's think then about why is the microbiome so important? What exactly is it that it's doing? So in the insight test, what the sorts of things that we can measure and address and understand are what it is doing. Now, this is a really, really key point because I still hear all the time people talking about the balance of good bugs and bad bugs. That's not really the case. That's kind of oversimplifying it a little bit. There are bad bugs. Those are the pathogens that we certainly want to keep very suppressed or keep out of the system altogether. However, it's a little more complex than that. You may have a particular bug that's doing a particular job, and I may have a totally different bug, but it's doing the same job. And that's what we would see with people all around the world. When I measured my microbiome compared to people, I grew up in rural Scotland in the country, right next door to a farm. I lived much of my childhood on a farm and outdoors and had lots of animals. My microbiome was very, very different to people who had grown up in urban Sydney. So, and you'll see that when you look at pockets around the world, people's microbiomes are as unique as your fingerprint. So it's not correct to talk about you need to have this particular bug or you need to have that particular bug. It's more about what exactly is your microbiome doing. So functioning is much more important. So when you look at these tests, what we can measure by, and that's why it's better to look at the genes actually than the species themselves. What is the capability of your microbiome to create things like butyrate? So that's one of the short chain fatty acids that's produced on fermentation. And butyrate's important because it fuels those cells lining the colon. And your ability to produce nutrients like folate, like vitamin K. We can understand its ability to produce a pro-inflammatory molecule, so something called LPS, lipopolysaccharide. So LPS has been identified in people at higher risk of cardiovascular disease. We know that it is produced more in people who have a very high saturated fat and a high animal food diet. So it may be, and this is what's fascinating to me as a nutrition scientist, that lots of associations that we've seen between diet and chronic disease, we've not really fully understood what the mechanisms involved are. And our understanding of the microbiome is kind of filling in some of those blanks. So sometimes there's a direct effect of nutrition on physiological pathways. Sometimes we're seeing now that actually it's via the microbiome that we're seeing these effects. And this is one example. So the production, you're going to produce more LPS depending on the bugs that you have and their capability of producing this molecule. If you have a very high saturated diet, it tends to increase the permeability of the gut to allow LPS to get up into the bloodstream. And then LPS is associated with these kinds of diseases. Now, whether it causes inflammation or whether it's causative in some way of the diseases, we don't yet know, but we certainly know that there's that association there. And we certainly know that this molecule is pro-inflammatory. We can look at your ability to produce branched chain amino acids. We can see high levels of microbe-created branched chain amino acids. So this is different to branched chain amino acids that are coming from the foods that you eat. We know that they're very important. For those of you with an exercise background, you'll know that those are important for muscle physiology and for muscle recovery after exercise. But this is slightly different. This is looking at specific branched chain amino acids produced on fermentation by the bugs in your colon, then being absorbed up into the bloodstream. And in this situation, high levels have been observed in obesity and in insulin resistance. So again, this is an observation. We don't know what's causative and not here, but it seems to be a better situation to be either just equal to or a low ability to produce these branched chain amino acids. We have, you may well have heard of this, is probably the best known one, trimethylamine or TMA. In the liver, it's converted to TMAO, and that's what's been getting a lot of headlines in this area because it's linked to cardiovascular disease again, and also to chronic kidney disease. So this may also be a mechanism by which we know a high meat diet, and particularly a high processed meat diet, is associated both with colon cancer and with other negative health effects. And we see this increased production of TMA with high meat and low fiber. Now, I put in that stuff about low fiber because we see time and time again, for me, the issue with meat is the way that we eat it. It's the portion size of the meat and whether or not meat is the hero on the plate, or whether you've got a lovely big selection of veggies or salad veggies to have alongside your meat. So we see again and again and again this protective effect of having veggies alongside meat. And this again is looking at that mechanism. So if you have low fiber and high meat, you've got this problem. You're going to have higher levels of TMAO. But if you eat your meat with lots of veggies, lots of whole grains, perhaps with some beans and cruciferous vegetables, so that's things like Brussels sprouts, broccoli, cabbage, cauliflower, those vegetables in particular are high in indoles, and indoles seem to reduce the production of TMAO. So just a really nice way of explaining why it's important that we have a big veggie intake alongside your meat. And this compound called IPA, indole propionic acid. This is a strong antioxidant. So we want to have IPA produced. It protects nerve cells from damage. It may protect against insulin resistance. And again, this is an effect of a really high fiber diet. So Things like rye bread have long been associated. We don't eat much rye. You can get rye bread in Australia, but I wouldn't say it's hugely popular. But it's incredibly popular in eastern parts of Russia. And rye has long been associated with digestive benefits and with good gut health. And so again, this might be the mechanism by which it's happening. It's increasing the production of IPA that then is having this knock-on effect on health. We can also look at the ability of the microbiome to break down fiber. And that's a good thing because fiber is the fuel that fuels the bacteria and the other microbes. I focus on bacteria because they're the major ones, but I'm reading more and more and more about the role that fungi, for example, are playing. So it is important to remember there are other microbes there. But we do want butyrate production. We know that a healthy microbiome is one that's producing lots of butyrate. So you're producing butyrate when you eat lots of fiber. So I do get very, very concerned with the current popularity of very low carbohydrate, very high fat diets. The keto diet is at the end of the spectrum of that kind of approach. They may well have therapeutic use in the short term, but long term, they're pretty much a disaster for a healthy microbiome. We need fiber. And fiber is found in plant foods, so you need to have a strong plant food intake, whether or not you also choose to eat animal foods. We can look at the ability of your microbiome to break down protein. Now, that's a bad thing. So, we tend to, protein is the macronutrient that has escaped scrutiny. It's kind of been the good guy for such a long time. And I would support that we need to have a good intake of protein. Protein can help with appetite control. It can help maintain your muscle mass. Of course, it's important for athletes or people trying to build muscle. It's incredibly important in our elderly to make sure that they are getting sufficient protein because we get less good at manufacturing it. However, there's a downside. There's always too much of a good thing. And if you're having too much protein because you're eating this incredibly high protein diet, you're drinking protein shakes at the drop of a hat, and instead of eating whole foods, that protein is not all going to be digested and absorbed, and you've got more of it going down into the colon. And when the microbes get hold of it, they increase the production of pro-inflammatory molecules. So we know that having too much protein, so some of it is escaping digestion, is not a good thing. And if you've got the type of microbiome, and you will have that type of microbiome if you're repeatedly following a very high protein diet, those microbes will have proliferated and they will be dominating, and you get an increased production of these kinds of inflammatory molecules. Your potential to degrade oxalates. So oxalates are intimately involved in the creation or the buildup of kidney stones. Of course, there's a genetic link here. Some people have a predisposition to kidney stones. So again, there's this interaction of your microbiome and genetics here. But if you have a low potential to degrade oxalates, you increase your risk of kidney stones. And then we have the production of hydrogen sulfide, which basically means smelly farts. So, and that's linked again to proteins. So those of you who go to the gym will know this. People who take too many protein shakes don't smell so good. And that's because of the production of hydrogen sulfide. So forgive me for having to talk, you cannot talk about bowel health and the microbiome without talking about poos and farts. So let's just dive straight into it. The truth of the matter is fermentation results in gases, and most of them don't smell of anything. But hydrogen sulfide does. And so if you have a high potential of your microbiome to create hydrogen sulfide, and that you pair that up with the kind of diet that supports those bugs, which are then creating hydrogen sulfide, not only is that fairly socially unpleasant, but it also impairs your gut barrier function. So it seems like a very small production of hydrogen sulfide is actually good, it's beneficial. But if you have too much of it, it's impairing that gut barrier function, and it seems to have some effect on mitochondrial misfunction. Fibers come into play in here, so specific probiotic fibers and resistant starch, that's RS, resistant starch, and fructooligosaccharides, which are particular types of fibers, seem to be protective, and they can reduce the production of this hydrogen sulfide gas. So another reason to make sure that you're having your proteins alongside plenty of fibers. Now, we've talked a little bit about how you develop your microbiome through your infant and your young years, but what about now? What can we do? What affects our adult microbiome? A big role here. You've already got a few clues there from what we've talked about. I thought I'd just throw in this slide because this was one of the most fascinating researchers that I interviewed on the show. And he's a guy called John Cryan. He's an Irishman. So he's got a much stronger accent than I do. So you have to tune into his accent. But if you just do a search on PubMed for some of his work, you can read about some of this. So this was really, really fascinating to me. And his question essentially is, could early life stress be involved in mental health? That's the question he's trying to answer. So his work so far has been in rodents. And what he has shown pretty fascinatingly is that if you take a young mouse or a rat and you expose them to trauma, I think mostly what they're doing is they separate these infant rodents from their mothers. If they get early life trauma in that way, what happens is that they have a measurable change in the development of their microbiome. And those changes, despite the fact that the mice and rats are put back into their normal lab rodent life, those changes to the microbiome actually last a lifetime. So the development is affected, and then that has lasted a lifetime. And then as adult mice or rats, they are then put into stressful situations to see how well they deal with it, how resilient these animals are essentially. And what they find is that those changes in the microbiome correlate with less resilience. So his question now, and he's trying to show this in humans, but of course it's a much more difficult situation to show in humans. He is showing that they have an altered brain-gut axis. He's showing that they have visceral hypersensitivity in the gut. So in other words, they're more prone to what in a human would be diagnosed as irritable bowel syndrome. They have increased systemic immune responses, so they might be more susceptible to autoimmune diseases and so on, and immune changes. But they are also less resilient. So the link he wants to show is, could this early life trauma be affecting your long-term risk of things like anxiety and depression and other mood disorders, based entirely on what happened to you early, and is the microbiome the mechanism involved? So for those of you interested in mental health, I strongly recommend, I've given you one reference there from his group, but online and you'll read more about this particularly. So let's focus more now on my particular area, which is diet and the microbiome. And I've already mentioned this magical word fiber, but just to illustrate, this is one of my favorite studies. It's a very small study, but it really is so nicely illustrative of the difference our diet and environment makes to our microbiomes. So this is now quite a famous study. It's a little bit old, it's 2010, but it's still a stand-up study in terms of really comparing the microbiome. And what they did was compare kids who lived in Florence in Italy, so in an urban, modern, developed world environment, with kids from Burkina Faso, which is in Africa. And you can see the images there are from their traditional village where these kids live. So you can already start to imagine what those differences might be. But in terms of their diet, the African kids were largely vegetarian because, you know, animal products are expensive, hard to come by. So they did have some animal products when hunting was successful, but it was a largely vegetarian diet. So you would imagine it's much higher in fiber, and it is. It's much lower in fat and lower in animal protein. The EU kids, the Italian kids, had about half the amount of fiber, and they had much more animal protein. They had much more sugar, much more starch, much more fat. So a very big difference. But you can also see there from the images, of course, their lifestyle is also different. These African kids are much more intimate with animals, with soil, with dirt, and far less kind of hygienic homes. But it was to their benefit when you look at their microbiota. So when we look at, don't worry too much about the individual species here, but there's been a lot of, particularly the early research, really focused on the ratio between bacteroidetes and the firmicutes, or the firmicutes, it's sometimes pronounced. Bacteroidetes and firmicutes ratio can be very, very different. So if you just look at the green to red ratio, look at the difference there between the African kids are up top and the Italian kids down on the bottom. Now, early research showed that that ratio between these two groups seemed to be important in terms of your propensity to become overweight or obese, insulin resistant, and so on. That's largely been disputed because it's a little bit more complex than that, but certainly there are still some key characteristics. And you can see that it's almost completely reversed. It's almost completely flipped. And when we look at their ability to produce short-chain fatty acids, you've heard me talk about butyrate, that's butyric acid, but these are the four principal short-chain fatty acids, and you can see the dark African kids and the light bars. it's different across the board. So they've got almost triple the amount of total saturated fatty acids, short chain fatty acids being produced. And then right across the board, including for butyrate, the African kids are producing much more of these beneficial short chain fatty acids. In terms of nutrition research now, for us, we have redefined fiber. So even when I first studied as a dietitian, 25 odd years ago, fiber tend to be talked about as one entity, and we still talk about that. Our Australian guidelines specify particular grams of fiber. They don't really delve down into the individual types of fiber. Redefinition and starting to understand that different fiber types, because there's hundreds of different types of fiber that we group together collectively. So you'll have heard of the term prebiotic. So essentially that means a selectively fermented ingredient that allows for specific changes. So it's not just willy-nilly any type of fiber, it's the type of fiber that has the ability to change the composition and or the activity of your microbiota. That's the gastrointestinal flora that confers benefits upon the host's well-being and health. So I hear lots of people just talking about any kind of fiber as being a prebiotic, and it's not. It has to have been shown to change the flora, therefore giving a benefit to the host for well-being and health. And then there's this new term MAX, which is nothing to do with the famous burger. It is microbiota accessible carbohydrates. So in other words, it's carbohydrates that lend themselves as fuel to the microbiota. So they're fermentable carbohydrates. And, we clearly see a difference in the microbiota in a MAC-rich diet compared to a MAC-poor diet. Traditionally, you'll have heard of soluble fiber, insoluble fiber, and then resistant starch. So CSIRO have been very great at sort of talking about resistant starch and about the importance of resistant starch because it's a primary fuel for the microbiome. So this little diagram is quite nice because it just shows what happens to these three groups of fiber as they travel through the gut. Now, I've heard it said that, oh, well, resistant starch is the important one because it's fuel to the microbiota. But actually, were you to have resistant starch on its own, you'd probably find yourself in trouble because it's highly fermentable. So it hits your colon, the bugs there are going to have a party and you're going to have a lot of gas production. And that's potentially going to be really uncomfortable, particularly in someone who's not used to having a lot of fiber. A lot of the soluble fibers also have prebiotic activity. So do some of the insoluble fibers. So it's not correct to say that insoluble fiber is just going unchanged through the gut. And if I just show you, first of all, what the three soluble fibers are. So we've got the three groups, So we've got soluble fiber present largely in things like legumes. That's your chickpeas, your beans and lentils, oats and barley, fruits and vegetables in the flesh. Your insoluble fiber plays an important role in keeping you regular, but some of them are still fermentable. So it's not correct to say they're insoluble. That's what you're going to get in the outer husks of grains, in the tough parts of fruits and vegetables, in the rough parts of things like Brazil nuts and seeds. And then resistant starch is your highly fermentable fibers. They're present in pasta salad, potato salad. So you cook and cool any starch. Some of it is going to retrograde and you'll get this resistant starch formed. You will still get it in legumes, again, are great for resistant starch. Bananas, when they're still quite firm. And in high amylose maize starch, which has been used quite a lot as a food additive in functional foods. Now, why I have the diagram up is because try not to focus just on the prebiotics. Those insoluble fibers are still important because they carry the other fiber types through the length of the gastrointestinal tract. So you can see right down there at the bottom, the insoluble fibers are the guys still left by the time it comes to right down to the rectum. But they carry the resistant starch and the soluble fibers through the colon, ensuring fermentation happens all the way to the end. And those of you with any medical knowledge of colon cancer know that's where most of these colon cancers are happening. So we want that fermentation to be happening throughout. The other thing they do is that actually the bugs start to ferment some of those insoluble fibers, and that releases the antioxidants. So we think of veggies as being rich in antioxidants, but actually whole grains are incredibly rich in very complementary antioxidants and anti-inflammatory compounds when they're teamed up with veggies. And what happens there with those that fermentation process going on is those antioxidants are released, and you get then helping to provide protection throughout the colon too. So in fact, we need this really good milieu of these different types of fiber, and that's what comes down when you get diversity. So fermentable fibers are playing a key role, primarily because they're creating this butyrate, which is what we want. And it's really important that we have them happening in the diet. And that's what most Australians are going really short on. So what happens if you don't have enough? Well, a low MAC diet's been associated with a decline in that bacterial diversity, which I started out by saying is really important. It promotes the growth of mucin degrading bacteria. So we've got these bugs, things like Akkermansia, one of the most commonly talked about ones. It's a specialist. It lives in the mucus layer. Now, it has a good role. Now, this is a really good example, actually. Remember when I said there's not necessarily good and bad? If you have too much Akkermansia, if it manages to get out of control because you're not eating enough fiber, then what happens is it's munching away at too much of your mucin, and you're going to get this degrading of your mucin layer, and your body can't keep up with replacing the mucin, and that affects your gut barrier function. Whereas if you've got just the right amount, if you've got a nice balance of Akkermansia with other bugs, then it's a good thing that it's breaking down mucin, because it is then encouraging your body to keep producing more, and you're getting fresh mucin being produced. So that's just a really nice example of why balance is so important. But what happens if you allow these guys to take control is you also get what are called mucin degrading generalists. So these are bugs like Bacteroides cacao that will actually will break down fiber if fiber is there, but in the absence of good fiber, it's going to start breaking down mucin. So you get this sort of bugs using a secondary fuel source, which is largely proteins like mucin or proteins that are coming in the gut. You have an irreversible loss of some bacterial strains. And again, the rodent studies have shown that this gets worse over generations. So you start to see, because you inherit your microbiome first and foremost from your mother, you can imagine that if you don't get some bugs, unless you're exposed to them in the environment somehow, you're never going to get them. So and that's exactly what we're seeing in modern Western societies. Our diversity is already lost. We've already lost some strains completely. And I think in the future, that's where probiotics might come into play, because you're going to have to put them back in there. The best we can do at the moment is try to encourage the growth and get an evenness with the bugs that we already have. You get specific immune pathways affected. So we can see that. We've got some good studies that you'll be able to uncover in patients with irritable bowel syndrome. There is also some work in inflammatory bowel diseases and what might be happening there. You get an increased susceptibility to infection. So why is it that some people seem to have a rock hard gut and be able to travel and not pick up many bugs, but other people seem to be able to pick up gastro at the drop of a hat? That might come down to your individual microbiome. And then you get this impact on the gut lining. So the epithelial integrity, that's a better term, I think, than leaky gut syndrome, and increased gut permeability. So you get this translocation I spoke about before of bacterial products. So I've talked about microbial diversity as being diversity, you need plant food diversity. So we know that 4 key groups are important. So we've got fruits, we've got vegetables, we've got whole grains, and we've got legumes. And the more, of course, allergies accepted and all of that stuff, yes, we have problems with some things, but for the large part, the more diverse different plant foods you can get, the greater the diversity of plant foods you're going to, the fibers you're going to get, and you're going to get a better diversity of bugs. There's another group of compounds, and again, until recent years, we really thought polyphenols were just antioxidants. We called them antioxidants. We encouraged people to eat them because we thought that they had an antioxidant effect once you absorb them up into your bloodstream. Now we know actually very few of them are directly involved or directly absorbed up into our bloodstream. Most polyphenols actually reach your colonic microbiota, and they are important sources for them. And either it encourages the growth of beneficial microbes, or they convert those polyphenols into more bioactive compounds that then have benefits on us. So we know that polyphenols play a really, really important role. So where do you get them? The foods are up on your screen now. We're talking about purple and red, blue foods. So things like berries are fantastic. Things like pomegranate, plums, they are in apples. So don't always think it's the colors. They are in green veggies too. Look for some very specific things. So red onions will have more than brown onions, for example. Leafy greens, so broccoli, curly, endive, spinach, and so on. Spices and fresh herbs are fantastic. Extra virgin olive oil, any of you who followed my work over the last few years know I'm a big fan of extra virgin olive oil. It's incredibly rich in polyphenols, as is olive leaf extract, which is a great immune booster, and table olives. And then we've got nuts and seeds. So this is again just showing that wonderful complement of different plant foods can be incredibly beneficial for your microbiome. I'm asked a lot about fermented foods at the moment, and the truth is there's not a huge amount of research there to back this up. But when you look at these foods, they are largely very traditional foods. They came about mostly because our clever ancestors worked out a way of making foods last longer. So usually they were created in order to improve shelf life before we had refrigerants. Many of them do contain some potential probiotics. And it may not be the probiotics themselves or the bugs themselves in some of these foods. It's actually, it might be the little acids, the organic acids that are produced on fermentation that we know to be good for our gut. And the fermentation may simply just make those foods more digestible to us. So look, all I can say at the moment is there's probably a benefit. We have got some studies emerging and coming out to suggest that they do seem to be beneficial, both for gut health generally, for the microbiome, and with this knock-on effect on overall health. So I think in general, these are really good foods to encourage people to consume if they enjoy them. Probiotics, I have a feeling that my prediction is if I can talk to you again in 10 years' time, we will be able to be much more specific about the probiotics we're using. The research is very mixed at the moment. We've got some great studies showing huge benefits, so you'll have no struggle pulling up some positive studies, but then we have other studies showing no benefit whatsoever, and even some showing detriments. So I think that the issue at the moment with probiotics is that if you think about how different our microbiomes are, how do you know which specific bug is going to be beneficial? Many of them are just 1, 2, or up to maybe 8 or 10 different bugs, whereas you've got hundreds of different species in your gut. So all I can say at the moment with probiotics is they do have potential. The sorts of things they might do is suppress pathogens, so it might be worthwhile. We have some clinically tested strains showing it can help to reduce travelers' diarrhea, for example, while you're traveling. They may be beneficial to take. I've just had to have a round of antibiotics after my head injury, so I certainly took some probiotics to try to help my gut to recover from having to take those antibiotics. So they may improve your gut barrier function. They may have a role in things like IBS or inflammatory bowel. They may help to promote immune function. So I was asked today about a specific probiotic that's being pitched as helping us to fight viruses, things like the coronavirus. So we need to see really clinical testing of those kinds of products to know whether we really are taking the right thing. They may help with the production of signaling molecules to the brain. So there are some studies underway associating or trying to look at whether probiotics have a role to play for people with depression or anxiety or brain disorders like cognitive decline. And they may play a bigger role earlier in the gut. So I certainly think that for babies born by cesarean or who have their eating upset in those first few years of life, we may well see specific probiotics being developed for those particular stages of life. I am going to throw this one cautionary word. This was a study by Israeli researchers that actually found that probiotics given after antibiotics inhibited the recovery of the indigenous microbiome. So in other words, they measured their microbiome, their own unique microbiome, looked then after what happened when they'd been given antibiotics, and then who recovered best. And actually in some people, antibiotics inhibited the recovery of their natural microbiome. Now that kind of makes sense to me in a way, because if it wasn't a good match for the bugs that are already in your system, it may be suppressing the regrowth and the recovery of some of those groups of bugs. Do fecal microbiome transplant. So this was using their own microbiome. So using that and putting that back into the system was the best way to get the recovery of their own microbiome. That's hardly surprising, but unfortunately at the moment it's not really too easy for most people to do. And as a positive note, for most people, if you just, as I've just had to do, take one round of antibiotics, you rarely take them. The evidence really shows that within 2 to 3 months, your own microbiome will recover, provided you have the right diet. So I think this sort of research is good to have as a cautionary word. And it is more, it's probably more applicable for people who have got to take antibiotics over a longer period of time for whatever their disease state may be. Those are the people we really have to focus on. We may have to put some bugs back into the system. So the quote from Dr. Elinav, who was the lead researcher on this, said, we need to wait for the generation of patient-tailored probiotics. So that's quite interesting. I see that we're going with probiotics, that we need to have a little bit better understanding of, I will be in a situation in the future, I hope, to be able to measure someone's microbiome and then be able to give them specific targeted probiotics that matches their microbiome situation. So to finish up, where do I think this is going? What is the future? We are definitely on that path towards personalized health. So I think that will include personalized nutrition and personalized medicine. But I urge you to just be careful with this. We are not quite there yet. Yes, we can now measure the microbiome. Yes, we can do DNA testing. So companies like Nutrigenomics that I've worked with, it is fantastic as a motivator to be able to tweak someone's dietary and lifestyle advice with these kinds of testing. But it really is tweaking, and it's adding motivation and so on at this point in time. We've got a lot further to go. And so there are unscrupulous companies putting the horse in front of the cart. So just be careful with that. Certainly the path we're on. We're definitely going to get more specific targeted probiotics. I think we're going to have huge dietary manipulation of the microbiome. We're seeing that, and we can do that already with the use of things like prebiotics and polyphenols. We're going to see those early life interventions to ensure people develop a healthy microbiome. That seems to be important for lifelong health. And let's see where we can take this in the future. But we've got an awful lot more to learn yet. So that is the conclusion of my presentation. Feel free to connect with me in any of these ways. I've given you my site, my social media there, but I'm more than happy to answer any quick questions now. And I know we've got a longer time. Sorry, I've filled up kind of most of the hour there with talking, but we do have a longer Q&A session coming up when I next speak with you. I shall hand back to Darren.
Good on you, Jo. Thank you so much for that. You know, I think there's a lot to digest. Digest, I think it's the right term for it.
Good word.
But yeah, look, a couple of questions have just flicked up. So I'll just, in the 2 minutes we've got left, I'll just ask them now. And as you said, we'll have a long time with the class. I know there are a few people have joined in here that are not part of the class. So you could think about joining the class if you would love the opportunity to ask Dr. Joanna more questions. Yes. But anyway, that's for a couple of weeks' time. A couple of things. I was interested. One of the questions related to, you spoke about recovering from antibiotics, and I know that you alluded to that on a couple of occasions. Any more comments?
Yeah. Look, I still think, and I'm there at the moment. So I just stopped taking my antibiotics a few days ago. I had to take them for a week following my head injury. And so really what I'm focusing on is what I would advise anyone else to is to make sure that you follow the kind of diet I've just been talking about. So I still think the evidence that I've seen on what happens, of course, when you take antibiotics, you're killing the good guys as well as the bad, but you don't kill them off completely. So it's not as if I've suddenly now got a sterile gut and I've got to start from scratch. What you actually do is you just suppress the growth of your bugs. You've sort of suppressed everybody down. And the strongest evidence I've seen is that actually provided you had a healthy microbiome to start with, your microbiome will recover from a singular round of antibiotics, provided you eat well. So if I now eat plenty of my beans and legumes and my whole grains and my diverse range of veggies and fruits and nuts and seeds, my microbiome is hopefully going to recover within a couple of months, and probably even shorter, given that, I know I had a pretty healthy collection of bugs there to start with. What I do think is a concern is if you have to take repeated rounds of antibiotics. So if you have to take, or you've got a patient who's having to take repeated rounds, then I think it is well worth introducing a probiotic. There are clinically tested probiotics though that have been used in this space, and that's what I would be looking for, rather than any old probiotic off the shelves. So I'd be looking for that clinically tested one, and it might be worthwhile doing a microbiome screen, doing a test to see what's happened and look at changes over time. And then really focusing on why diet is so important and lifestyle. Forgive me for not throwing in much about the lifestyle. There's so much to sort of say there in a singular lecture, but remember that things like sleep and stress and exercise, all of those aspects that we know, alcohol intake, all of these things, smoking, they all also have effects. And what's nice though is it's very aligned with what you will have learned about what is good for heart health, what is good for brain health. These things are also good for gut health. So it just kind of shows us that there is this healthy lifestyle that includes a healthy diet that actually is good for all of these different bodily functions, which makes sense.
Last question then. You mentioned that you don't like the term leaky gut. Now, I know you use a couple of other terms like, you know, permeability and the like. Can you just elaborate on that?
Yeah, look, I guess I don't like the term leaky gut because it's been so misused. So it's been used over the years, and I've used it myself. I remember using it when I was speaking to a group of doctors in Perth many, many years ago, and I was talking about the science behind A2 milk. So I wasn't promoting A2 milk. I was just trying to explain what the science was, which was my role at the conference. And a doctor who came on after me said, well, that was a load of nonsense. You can throw that presentation out because leaky gut syndrome doesn't exist. So I feel quite justified now in being able to say, I can see it because we've got the technology. The gastroenterologist I worked with on Catalyst has developed the technology to be able to visually view it. And he's showing it very clearly in patients who have got inflammatory bowel disease and are in remission, and he's now starting to show it in some patients with IBS. The reason I don't like leaky gut is because I think to the public and to some less qualified, shall we say, people giving health advice, they're sort of explaining it as if, you know, I eat a steak and I get molecules of food getting up into the bloodstream. So it's not as if you have giant holes in your gut. It's just that, and it may not be all the way through to, you know, it's not as if things are getting all the way through to the bloodstream. It's more that you don't have as strong a gut lining. It's almost like, you know, your skin's getting thin and more things are being able to absorb through your skin. The same is true in your gut lining. So you're not producing mucus correctly. You've lost some of the linings. Bacteria are managing to get in a few layers. They're not supposed to be, and then that's triggering an inflammation, and it's the compounds that are then produced through inflammation that are getting up into the bloodstream and having an effect elsewhere. So I think we just need a slightly better term. I don't say we have to not say leaky gut at all, but I just think it's been so misused and misunderstood that we perhaps have to talk about it in a different way to ensure that everybody in the medical community as well gets on board with understanding exactly what's going on.
Good answer. All right, that brings us to the end of our time for tonight. But for those in the class, save your questions because we'll be having a Q&A. The next session will be more informal and we'll just be able to pick Jo's brain and she'll be able to set us on the straight and narrow and give us all the good oil. So, so that'll be in a couple of weeks' time. I'm just going to finish the recording now.